All right, hello, it's me again. Welcome back. I hope you guys all had good lunch. Today, now for our afternoon hands-on section, we're going to be going over model generation. I wanted to give a brief outline because compared to the last one, we will have two parts. The first section will be model generation with NeuroDOT and NearFaster. It's going to start with like a brief 15-minute talk overview, and then the majority is going to be spent in the hands-on section. I'll be leading this portion, and then the next portion after that is going to be model validation using reconstructed data, which Vito over here is going to be leading. Again, it's going to be split in half for a talk and a hands-on section that she's prepared. So for our learning objectives, after this section, you guys are going to be able to understand the pieces needed and steps used to create a light model, aka the A matrix or Jacobian. You're going to be able to position an array on a head mesh using the AlignMe app, and then also validate the A matrix by visualizing reconstructed data compared to fMRI data. So a brief overview of image reconstruction, as we talked about with Adam earlier. In light modeling, our goal is to model how variations in the light level correspond to changes in optical properties. And this relationship we can describe through an equation, y equals ax, where y is our measurements from our source detector pairs, A is the sensitivity matrix, and then x is our changes in absorption. And essentially, in light modeling, our goal is just to make the A portion of this equation. So what is the A matrix? A is the sensitivity matrix. It's the Jacobian. Those names for it are commonly used and interchanged throughout the field. It's a model that relates differential changes in light measurements to differential changes in internal optical properties. And the sensitivity profile can be visualized by making a flat field reconstruction of the A matrix. So that's just one of our tools to visualize where we're getting sensitivity to, and that is primarily within the regions that are colored in red on this flat field reconstruction plot. So in terms of pieces, what do we need to create A? Firstly is our anatomy. We need an anatomical head model, which usually is a finite element mesh, which contains the boundaries of the different tissue types within the head, and then also accurate locations for all the optodes relative to the head surface. And we'll get into what we mean by accurate locations for the optodes in just a little bit. In addition to the head model, we also need optical properties for each of our tissue types, such as absorption, mu-A, scattering, mu-S, and then the index of refraction, which is n. And then lastly, we need the wavelengths of light that our system is using, which is always dual wavelengths. In terms of our pipeline for model generation, in our big figure, it's just the part that's circled in red. We start with MRI acquisition for participant-specific light models. We then will perform image segmentation with FreeSurfer if necessary. Or if we're starting out with an atlas, we will already have a segmented volume to go off of. In terms of the later parts of the pipeline, we'll have mesh generation and optode placement, which is repeated for both a low density and a high density mesh. And then we'll calculate the sensitivity and then validate the sensitivity after that. So in terms of head model generation, just generating our model of the head and brain, we use, here in our lab, we use FreeSurfer. So we'll unpack those FreeSurfer outputs and then segment the head volume with NeuroDOT. These two steps are really only relevant for non-atlas-based head modeling. So if you have a participant MRI, this is what you'd be doing in that case. And then after that, you'll generate your computational head mesh and then align your array. So we use the segmented volume to make the head mesh. And then in our lab, we use an app that Abby McGee developed to align the array with the mesh, and that app is called AlignMe. So we do have light model generation after that. So this is when we go into calculating how the light diffuses through the head, and then we validate and visualize the sensitivity profile. So that's what Joe was talking about the other day and Adam was talking about this morning. We generate Green's functions for each source, then generate Green's functions for each detector, and then we combine the Green's functions for each source detector pair. And all that is just multiplying our Green's functions for a source by the functions for the detector. That gets the source detector pair. And then after that, we can visualize our the aspects of our sensitivity matrix. I primarily use two visualizations. The first is for single source detector pairs, which helps me know that my measurement list is correct and the left side of the cap corresponds to the left side of the head. Same with right, top, and bottom. It's just good for spatial organization. And then the flat field reconstruction, which tells me, am I covering the areas of the brain that I actually want to with this model? Because if we're not, then we might need to regenerate a model because we're not looking at the right areas of the brain for our paradigm. And then lastly, we'll create a field of view for the sensitivity matrix, which is useful in processing and visualization for just cropping our data just to what the cap is actually able to see, as opposed to just showing the entire brain when we don't actually have data from portions like that. So yes, we have a question. Yes. So if you're making, the question was, if you are making an atlas-based head model, which of the steps in this light model generation slide will you be doing? The answer is all of them. Yeah, none of this changes if you're using atlas versus participant. But the slide before it, it's just the free surfer stuff that you will be doing only if you are doing a participant specific. If you're doing an atlas-based, then you start with the mesh generation.
Thank you.
No problem. So some of our key steps in model generation, we start with the image segmentation. So for today, you guys are going to be working with my head. I have not defaced it because I can consent. So all of you guys get to see my brain and head. So image segmentation with FreeSurfer to start. That's how we got this wonderful segmented volume. We'll then do optode positioning with AlignMe, calculating the sensitivity with NearFaster, and then validating the sensitivity with some NeuroDOT visualizations. Yes, question in the back. 1 sec, wait for the microphone to get to you so we can help the people on Zoom.
So when you place the optodes on the mesh,
Yeah.
so do we need to align like Nasian or Inian?
That's a very good question. It depends on the intensity that you want to go for your alignment process. For example, I've done some pipelines where we auto-register a digitizer probe array to the head, and for that we do need to use the Nasian and Inian because we're aligning everything in that sense. Additionally, I'll get into this in a little bit.
Okay,
if you're scaling the mesh to participant-specific values, you will need those landmarks in order to generate the affine matrix to go from your atlas to your participant-specific values to just stretch and transform that mesh.
Okay, thank you.
No problem. Alrighty. So our focus for today, the first is going to be generating a pad file, which is not something that I've talked about, but I'll get into that in just a moment. Then we'll go into optode positioning with AlignMe, and then validating the sensitivity. The hands-on section after this talk will be on these topics using a WashU adult 24x28 visual pad, the same one that was used for the retinotopy data that we were looking at in that last section. And then we'll generate a light model using that above pad file and an MNI atlas. So that brings us to the question, what is a pad file? A pad file refers to NeuroDOT's representation of the imaging array. This is typically named info in the code because a pad file is essentially just a bunch of metadata about your array. Pad files contain 2 key fields, info.optodes, which is your optode locations in 2D and 3D. These are the necessary inputs to build any pad file. You have to have both of them, and from that, all of our NeuroDOT codes can just generate your pad file for you. We'll go into this in the hands-on section. And then lastly, we have info.pairs, which is your measurement list. Your source detectors, the pairs of those that are all together, wavelengths, and everything else. If you guys have any more questions on what is the pad file, how is NeuroDOT's data organized, highly recommend checking out the NeuroDOT data organization tutorial. Also, Mike over here in the second row has made fantastic YouTube videos, so if that's more your speed for internalizing information, check out the NeuroDOT YouTube channel. He's done a really amazing job with putting together all of our PowerPoints and scripts into a video format.
To find those, do we just search NeuroDOT YouTube?
Yeah, so the question is to find the YouTube videos, what do you search? Yeah, just search NeuroDOT YouTube. And you'll be able to find it. There is another channel on YouTube named NeuroDOT. We could not get the name for it, so if you just search NeuroDOT and look for the first thing that looks like diffuse optical tomography, the other channel doesn't have any videos, so ours will show up. So now I'm going to walk through the pipeline. For this walkthrough, I'm going to be using my head with a Lumo visual pad. But for what you guys are going to be doing, we're going to be using the WashU fiber-based pad. So we'll start with the segmented volume of my head. You can see that there are different colors corresponding to different tissue types. We make a 5-layer model with scalp, skull, gray matter, white matter, and CSF. Then we'll generate a low-density mesh and visualize. In terms of mesh generation, there are 3 parameters that I want to highlight. The first is the facet distance, which is your error tolerance at the boundary for node positions. And so that's just like your points on the mesh, how like far away are they. We typically, for low-density meshes, are set that value to 2.0. We have the facet size, where we're making a mesh out of like essentially polygons, polyhedrons. And so the size of the surface polygons, that's what facet size mediates. We want to keep this as small as possible to ensure a highly faceted mesh. So that's why we set that to 0.8 for default. And then the cell size, which mediates the size of the internal tetrahedra. And this is the parameter that we primarily tune to differentiate between low-density mesh and high-density meshes. For high-density meshes, you always want to ensure that the number of nodes within your mesh is less than 1 million. If you have more than that, it can cause issues with the near-faster code. So if you're generating meshes on your own, whenever you get to that HD mesh generation part, just take a look at the number of nodes inside your mesh, make sure they're less than 1 million, and you'll be good to go. And our defaults for that are set to 1.0. So after that, we'll load a pad file in Visualize. We can visualize it in 2D. We can also visualize it in 3D. And then we can visualize it in space with reference to the mesh. And so a real useful part about visualizing your mesh and your array in space together is sometimes your array will exist inside the mesh because we're just all in coordinate systems. We're just in 3D cubes. So sometimes where that array is positioned is not actually relative to any mesh. It's just in space. So that's the whole point of optode positioning, is like getting your array to be on the surface of the mesh as opposed to some other place in space. So now I'm going to give a quick overview of our AlignMe app. We'll go through this more in depth during the hands-on part. The use cases for our app depends on the type of the head model that you're building. If you're building through a participant MRI, that's the best possible case because it's accurate to your participant, and therefore no atlas scaling is needed. You can also use an unscaled atlas, which is very useful for group analyses and group level models. And then AlignMe is really cool because it posits, it has a middle ground between those two, which is atlas scaling. We can use 3 different types of values: head circumference, nation to Indian distance, and then tragus to tragus, so ear to ear distance to scale our mesh. For this talk, though, we're just going to be using a participant MRI. And so another cool thing about NeuroDOT and AlignMe, we're system agnostic. It doesn't matter if you have an Artinius, NIRX, Lumo, or home-built system. We can work with all of them. So a brief overview on the GUI's face. We allow for atlas scaling through this top box highlighted in red. Head circumference, C-line, and Z-line distance are our metrics that we can use here. You can use any combination of them for your usage. We also have the positioning box, which we start with the scale, which increases the size of the array, and this is super useful for spring relaxation. The way we do array placement is we model the system of optodes and nodes on the mesh as a set of springs. And so for those of you who are into physics, when you increase the energy of that system, you can then let it relax naturally onto the head. So that's how it works. I'm not going to go too much into the math on that. But essentially, one of our first steps in the model generation process with AlignMe is to increase that scale to 1.1 or like 1.05, depending on how large your grid is. We also, in this box, can translate, rotate, and flip the array around axes, which is useful if, let's say, your array is upside down to start relative to the mesh. Always easier to flip a set of points that's less like an array than like a large number of points as in a mesh. It's just computationally more easy to do. And then lastly, we have a UI on the right-hand side of our app for photos. This is super useful for ensuring that your cap placement is more in line with what was happening in the clinic. So when the cap was actually on a participant, we want our model in computer land to be as close to what we saw in the facility as possible. And so taking photos of the cap fit and using that to position the array based on landmarks is super, super useful. We can rotate photos if they end up being upside down, and then we can seek through a bunch of photos, because if you're taking photos during cap fit, you probably want to have multiple views, side, back, front, top, down, etc. Zoom question, yes.
So there's a question from Zoom. Can I use NearSight Atlas Viewer derived 3D coordinates as the 3D positions for the pad file?
Absolutely you can. So yeah, you can use any sort of 3D positionings, whether they come out of Atlas Viewer, if they come out of Digitizer, if you just manually set them yourself, or if they were derived from your system. All of those, as long as they are 3D set of positions for your optodes, can be used. For Atlas scaling, we have our 3 values: head circumference, left to right, preauricular distance, which ear to ear, and then nation to Indian, front to back. The more metrics you use, the more accurate your Atlas scaling will be, but you can't scale the mesh more than once. So if you need to redo anything, you'll just hit that reset mesh button, and then reset to the initial scaling, input your values again, and then scale the mesh. If you do choose to scale your mesh, AlignMe will output an affine matrix, so that way you can go from your scaled space back to your Atlas space for group analyses. This is really helpful if you have multiple participants and you're scaling to each individual one. A brief overview on translation. We can rotate the array. We can translate the array forward, backwards, up, and down. We can use photos, where we use these top bars, top buttons up here to rotate and seek through our photos. We can also change the view of the mesh and array to match our photos as well. All of these are pretty simple to do with AlignMe. As for our relaxation process, I alluded to this earlier, but we treat our array optodes and the mesh's surface nodes as a system of interconnected springs. That's why we scale the array up in AlignMe to introduce energy into the system, and then our algorithm relaxes the array onto the surface of the mesh by minimizing the total energy of that system. And these are the 2 plots that will pop up and auto-populate and update as the relaxation process is going. So if you hit that relax array button in the bottom left-hand corner, then these 2 plots will start to pop up and populate as that process occurs. And then when you're done, you'll end up with optodes, red for sources, blue for detectors, on a mesh. If you're looking at the middle image and you see the pink and light blue, those correspond to source number 1 in pink and detector number 2 in blue. It's just a visual way for us to show the first optodes for sources and detectors in that measurement list. So you've made an A matrix, you've positioned your array, you've calculated your greens functions. Now what do? Validate the sensitivity. I use 2 different things: single source detector pair, which helps visualize if the A matrix is out of order, and then the flat field reconstruction, which is useful for determining whether or not your system is in the right regions. So for a single source detector pair, the useful thing to do is to first visualize your 2D placement of all of your array, all of your optodes, and then compare that, use that to choose 2 optodes, a single source and a single detector, and then visualize the sensitivity for that measurement pair on volumetric visualization, such as in plot slices. And if they match up like they do here, then you know, hey, this is all good. I usually like to use 2 or 3 different single source detector pairs just to validate different portions of the cap if it has a larger area of coverage. Lastly, the flat field reconstruction, or FFR. It shows the sensitivity profile of the A matrix, and it shows that on top of the anatomy that it was created on. What that means for scaled Atlas scaling versions of model generation is, if you start with an Atlas and then visualize your scaled A matrix on that same Atlas, it won't match up. You also need to scale that Atlas the same way. So it's just making sure your A matrix and the model it's in and the anatomy you're visualizing it on are always in the same space. So that brings us to the tail end of my talk. We'll get into our hands-on section in just a moment. After this section, you guys will be able to understand our pieces needed and steps to use to create the A matrix, such as the anatomical head model, the array info, and the optical properties of the tissue. The steps, image segmentation, optode positioning, calculating the A matrix, and validating it. You'll also learn how to position the array on the head mesh using AlignMe, using atlas scaling, translation, rotation, or scaling the array, and then participant photos to validate the positioning. And then lastly, visualizing the aspects of the A matrix to validate that it's been created correctly. This could be done in single source detector pairs or flat field reconstruction, and then what Vita is going to be talking about later, which is visualizing reconstructed data and then comparing that to fMRI data. So a brief teaser for Vita's section. She uses a dice coefficient, which is just a measure of similarity between two sets of data, and it's used to assess overlap. So that true color and overlap parameters that I was highlighting at the end of the visualization section, we're going to use that to compare DOT and fMRI data. And so this is an example for an alternating checkerboard task where not quite retinotopy, it just flashes on the left or the right as opposed to a rotating wedge. So now I'm going to have everyone open up. We got a question in front. We got a mic coming down for you in just a moment.
Thank you. So I'm better starting with big picture things. Some of the pictures that you showed look similar to what we did in the visualization session
Yes.
last. What is the difference between what we're doing?
Totally. So the visualization section was just me highlighting a bunch of visualization functions that NeuroDOT has,
Okay.
which are used at multiple different places within the pipeline. It's not like we're going to use volumetric visualizations only in one specific place. They can be used at multiple steps to visualize the data in different ways.
Gotcha. So some of the stuff we learned then we'll be utilizing now?
Exactly.
Okay,
So
perfect.
what you've learned earlier is like, hey, these are the pieces, these are the functions that I'm going to use, and now this is the application of that into a pipeline. No problem. So yeah, let's pull out our MATLAB. I'm going to get set up on my end, so just give me a quick moment. So again, make sure that everything is on your path. I will have helpers around. If there are any questions that you think are too low level to like broach the entire room, people are around to like individually help you as well. So don't feel afraid to just be like, hey, I need help with this tiny thing, but I don't want to like stop what's going on. We got a lot of people in here who are very experienced with NeuroDOT. So I'm going to clear my stuff. And then hands-on section 2. Alrighty. So the first thing is pathing. I like to set an output directory for everything. So I'm in this folder right now. I'm just going to create a new folder for all of my outputs. I highly recommend if you're generating light models using NeuroDOT, you always create a folder for your light modeling outputs and work within that one for each individual light model. It'll make your life easier. So we'll say demo will be my folder. I'm just going to quickly add it to my path. And then I'll do this, replace my path right here with yours, and then add demo. So if I run this section, it takes me into my demo folder, which is currently empty. So the first part of this is we're going to build a pad file together, and we can build a pad file by starting with our imaging data. So I'm going to load in some data using that was collected using the 24 by 28 visual pad. So that's what this section is here. There is no, I see. I have my pathing wrong because I'm on a Windows computer here. So if you end up with the same error like me, you will need to just reverse your things right here. No such file or directory. So that is because I am not using the correct thing. So I'm just going to go into NeuroDOT main, data, and then copy this path right here and replace like this. And so this should mean we're able to load our data now, and now we have DOT data. I'm just going to scroll back up and go to my output directory again, so I'm not saving anything into the NeuroDOT toolbox. So we're going to start with our optode positions. So we can get that from our DOT data right here by getting our, looking at the info structure. So we have now created a grid structure. This contains our source positions in 3D, detector positions in 3D, and then those same positions in 2D for sources and detectors as well. Then we'll set some initial parameters. So we'll set our lambda values. We can get this from the system itself, or you can input them manually. Our modulation frequency, which is just CW, and then we also input a cap name as well, which I've named for all of you here. And then we'll run the generate pad from grid, which is just NeuroDOT's function to create your pad file for you. And then after that, once that's done, very easy process, we can visualize our layout of our pad file. So if I run line 68, we'll get a 2D layout, and we can see our grid. Blue is detectors, red is sources. We can also visualize in 3 dimensions as well. This is one of our functions, and then we can visualize our 3D layout using a different function called draw foci. And that would look something like this. And so you can move them around as needed. Lastly, what I like to do as well for some sanity checks is I'll visualize the 2D source detector separations and then the 3D ones as well as histograms, just to see what my measurements are looking like in terms of distances. So we can see that. And so these will always differ depending on your system. And then lastly, what I think is the most useful one is the source detector separations and nearest neighbor plots. You're organizing your nearest neighbors based on source detector separations. And this is a really useful plot in terms of debugging when you're making your light model. There are some issues that happen if your first nearest neighbors are not set appropriately. So for example, if I were to have my first nearest neighbors include these values over here, my spring relaxation function would probably mess up a little bit. So it's important to separate your short measurements from your medium distant measurements. And we have a question over here.
I just have an individual issue.
Oh, yeah, totally. No, just an individual issue. Morgan or Emma, could you help? Thank you. Awesome. So we'll continue on here. The last step afterwards is to save our pad file and then just clear the workspace of stuff. So I'm just going to run this entire section so that way we just have our output directory left, and now we can go on to generating our A matrix. Yes?
Yeah, I don't know if it's important, but I'm not necessarily understanding of like what's your spring relaxation. Can you maybe elaborate on that, or we can talk afterwards if it kind of diverts from the.
No, that's totally valid question. So for how why the spring relaxation function is necessary is head curvature and pad curvature don't always line up one-to-one. You may position the pad where like every single optode is very close to the mesh, but maybe 1 or 2 aren't actually touching. And in order to like accurately model the source and detector like positions, they all need to be physically touching the mesh in order for NearFast to do its thing. So the way we do that by modeling everything as springs is it ensures that at the end of the relaxation process, each individual optode is just touching on that mesh. It's easier to model it as springs instead of like moving each individual optode one by one, because then you're conserving the topology of the grid itself. No problem. So we'll head into our light model generation section. I'm going to run these lines to get our parameters set up, and then I'm going to load the participant-specific volume. This is the file that I was requesting all of you guys to re-download the box folder for because I needed to re-upload that. So here we can see my brain and my head, and we can hit Q to close out of plot slices and move on with our low-density mesh generation. So right here, I'll just run this section. And this is the reason why I like having my own folder. As you can see, as soon as I hit that button, 2 files popped up in here. And so whenever you're generating a mesh, NearFast and Segel mesh generator will write files to your folder. So it's easy just to have your own separate light modeling output directory. And so we can see a look inside of our mesh, and then we can see the full. See, doesn't this look directly like me? All righty. So now we'll load in the pad file that we just all created together. And so then we'll visualize it in 2D and 3D, and with the mesh as well. So we have our 3D, we have our 2D, and then we have the mesh. And if I'm looking for it, I can't see it, which means it must be inside the head. So I'm going to close all of the figures that I have open right now, just because AlignMe is a GUI, and it is easier to run when there are less figures open. If you have older laptop, just conserving computational power for that reason alone. So this next section is the AlignMe section. We start out with Atlas fiducials for our Atlas. I'm not using an Atlas, so these won't actually get used. But these are incredibly important if you're doing any sort of Atlas scaling, because it tells us where the landmarks are on the head. And then in terms of input, we have our optode positions in T-pose. We have our low-density mesh. We actually have the pad file itself, visualization parameters, the number of sources, and then if you were to have photos of your participant, you would set the path to those right here, and then the image type to JPEGs right here. I will note, if you take photos on an iPhone, please make sure that they are saved in JPEG form instead of .heic form, because MATLAB does not like Apple's HEIC, like high efficiency format for images. So just try to keep them in JPEG or PNG if you're planning on using any sort of participant photos in MATLAB. So we'll run this section now. And we have AlignMe open up. Another thing about AlignMe, it's just a little idiosyncrasy for any of our Linux users out there. I primarily use AlignMe on a Linux computer. And a weird thing about MATLAB App Designer and Linux is you can't actually type in any of the edit field boxes on a Linux device unless you touch the top bar of the app. It is a feature of MATLAB. So just a warning for any of you Linux users, if you're trying to type in AlignMe and you can't, just touch the top bar, wiggle it around for a moment, and then you can. So the first thing I'm going to do is try and find my array. This reset drop-down box at the very bottom is super useful for that. So I know I'm using a visual pad, so I want to set it behind the mesh. So I'll click that, and then what I can do is I can turn my mesh and see, okay, my pad's back here. First thing I'm probably going to want to do is rotate the pad. So we'll rotate this 90 degrees in the X direction because of the right-hand rule, and now we have our pad looking like this. And so I can go to a better view. This is a top-down, or I could go to the back view, and I'm going to position my array via the Z-axis now. So I'll modulate this, and usually what I do for AlignMe here is just play around until I find a good positioning. And so for this array, I know that the bottom row of optodes sits just above the top of my ears. So right now it looks like it's a little bit in my ear, so I'm going to keep raising this up by factors of 10 until I get to a good position. So I think I could raise it up a little bit more. And then I'll probably just raise it up by 5 now just to get it to here. I think this is a solid positioning of this array. And so it looks like it's on the head, but if we were to change our view, we would see, okay, it's a little bit far away. And then I remember, oh, I need to scale my array up, so I'll just hit 1.1 for this. I'll bring you back to this view right here, so that way you can see more directly what happens here. I'll put it back to 1.0. So you see it gets lower, and then as we scale it up, it just increases the distance between all of our sources and detectors. So we're here for aligning and relaxing the array onto the head. We want it to be around a centimeter off of the mesh to then relax onto it. So I know I need to move this a little bit closer. So I know I need to modulate dy now. So I'll move this down to, let's say, 143. That's a little bit closer. I think I can go even further than that, so let's try 123. And that's pretty damn close. So we're going to start with that. So the last thing I like to do is just visualize in terms of all views, just so I know what I'm looking at. And then I will hit the Relax Array button. And so what you see here is our plots start to show up. You'll see the starting parallel pool. This is one of the reasons why we had you download the parallel computing toolbox. It's a computationally expensive algorithm, so having more threads to do that on helps. So you just got to wait for a little bit for your parallel pool to start up, and then you'll see as these figures start to update themselves. So I'll just have these both go side by side so you can see. One here, and then one here. And so this top plot on the top left is your max tries and then on head. So the blue line indicates how many optodes are actually on the head, and the red is like how many tries it took to get it there. You have your energy plot in the top right, time per loop in the bottom left, and then average distances for nearest neighbors as well, with the energy of the configuration in green. And so we just wait, we let this update, and then there will be a dialog that pops up in the bottom, your command prompt once everything has finished. And so just like that, it'll say energy difference below threshold, terminating loop. It took a minute to do this. Sometimes for larger pads, it'll take a little bit longer. Additionally, a more complex thing that happens is sometimes pads are incredibly dense and you need to split them into different portions. So for example, if I had a system that had a left temporal pad and a right temporal pad, it would be easier for me to treat those as two separate pads, relax them onto the head individually, and then combine them once I've gotten those relaxed optode positions to then calculate my Green's functions. So now that our mesh is relaxed, if we go back into Align Me, there will be a dialog box that says mesh is now relaxed, and then you can hit the Save Alignment. I will issue a warning. If you double-click the Save Alignment box, it will error. If you double-click this OK thing, it will error. Just be patient with Align Me. It can get a little testy if you repeatedly click your input boxes. And then, when you're done, that section will pop up a relaxed position of the head. And so something you can do here, just to look around, is move this around. And then if you want to be extra about this, you can actually zoom in. And if you look inside the head, you can just barely see all of these optodes inside the head. One of my validation steps, if I'm like really being super attentive to everything, is to look at the outside, look at the inside, and make sure that everything shows up there. There we go, back to normal. So once we're done with this, we can scroll down. We have a question in the back, yes.
So when you're positioning the Z values, like you're increasing like manually, right?
Yeah.
So for a first-time user, how do we know like we are doing it right? And would the photos during the session help us? Like, no?
The photos during the session would definitely help you, just for getting the positioning. And then the last thing is just, instead of it touching the mesh before you hit the start button on the relaxation process, just have it a tiny bit off the mesh. But other than that, using your photos to position the array is the best way to go. So for this next section of the script, for ease of access, I have removed some sections that are extra long. The sections that I've removed are generating a high-density mesh, relaxing those optodes with Align Me onto the high-density mesh, and then the prepare step for near faster, which essentially takes all of the relaxed optode positions and your mesh, pushes them together into one single variable that near faster uses. That entire process for those 4 steps takes anywhere between 15 to 30 minutes, which is just time we don't have here. So I've done all of that on my end, and we are going to load in those relaxed optodes in a high-density mesh right here in line 252. So we'll load that in. It might take a moment. It did not because my computer's fast. Let's go. So now we can visualize our high density mesh with relaxed optodes. Here we go. Evaluate this section. And so if I were to compare this to our previous one, so this is our low density mesh. I'll just set the view on that one to the same view so we can compare. We have our low density mesh, and then we have our high density mesh. And you can just see that the faceting of the surface of each mesh is different. We have a lot more facets on our high density mesh. And then a final visualization check that I like to run is I'll cut a quarter out of the mesh and then visualize that. And this just helps me see if any of the optodes are stuck fully inside the mesh, and if I just missed that. But I can see here that they're not inside the mesh. They're just barely touching the surface, and that's exactly what we're looking for here. So now we can calculate our sensitivity profile. I'm going to move this a little bit lower so we can see some important parameters to note. We have our voxelation resolution in G. We also have our voxelation resolution in millimeters. And then we have labels for all of our optical labels for all of our regions. So that five-layer segmented volume I showed earlier, we're just setting those labels here. And then for each of those tissue types, we have our baseline absorptions, our scattering coefficients, our index of refractions. We also have the 2 wavelengths of light that we use here. And then we include the number of sources as well. And if you did atlas scaling, your affine matrix will get saved into this flags section right here. And then lastly, we use our function make a near faster to generate our A matrix, actually calculate the Green's functions. So what I'm going to do is I'm going to run this entire section, and you'll see that there is a little dialog that shows up in our command window that says making Green's functions for wavelength 1. And that'll show up, and then you'll see making Green's functions for wavelength 2 as that one started. And then when it finishes, you'll get that simulated light fall off that Adam was showing earlier as a figure to show up. Yeah. Could we get a microphone up here in the front?
So how do you determine the baseline optical properties for each of the layers?
Yeah, that is a great question. How do we determine the baseline optical properties? That is usually from previously established papers from the literature. You could probably run tests to measure those yourself, but I personally don't know how to do that, so I'm not going to advise that. But this is the most like mathy computational step for all of this. The other computationally heavy parts is just like building the high density mesh and then moving sources and detectors. So this might take a little bit longer because it's graphics for AlignMe move a lot faster on laptops, but actually calculating the Green's functions moves a lot slower compared to if you were on like a high performance computing server. So this may take 2 to 5 minutes. So if any of you guys have questions about anything that I've gone over until now, happy to field any questions you guys have. Could we get a microphone down for you real quick?
It might not be necessary for everyone, but
All good.
Okay, so I have a Mac, and I kept getting the security question.
Yes.
And Emma helped me earlier override it, so I just like did it again for this. Is there a better way to do that, like where I can just change my settings to where I don't have to do that every time, or just do that every time?
As far as I know, to get rid of the Mac permissions thing, I think you do have to do it every single time. It's an unfortunate thing with Macs.
No worries.
Yeah.
Hey, Ari, I was just going to mention some of these steps that you left out, for example, creating the high density mesh. I think you described those in the PowerPoint tutorial for alignment, and it's actually quite well done in that tutorial.
That is correct, yeah. Thank you
Or
for mentioning that, Mike. All of the steps that I've omitted in our light modeling tutorial, we have several light modeling tutorials spanning a variety of different types of models you would like to make. All of them do contain the left out sections. And additionally, I have created a version of this script that contains the omitted sections as they're meant to be, and I will upload that to Box right after this section, so that way you guys can have your own version of this script that contains the entirety of the full pipeline.
While we have time, in the PowerPoints, is it obvious, like, what we, like for our, let's say we want to go home tonight and do this on our own data.
Totally.
What pieces of it we change for our data versus stuff we like keep because that's like the toolbox?
Yeah. So in terms of the steps, everything will be the same unless you're splitting the pad up. In terms of what's different, it really depends on two things. If you're using a participant-specific volume, if that is different, then or an atlas, you will end up changing this line right here to just load in a different volume.
Okay.
And then the other difference would be your pad file. And the only thing that would change there is lines 168 and 169 to load in your pad file of choice.
Okay, dumb question.
Of course.
So we don't have MRI data, so we're going to be using atlases.
Totally. You can use
Where
one
do we find those? And
in
like
the
what
NeuroDOT
exactly
toolbox.
do we put?
So in NeuroDOT, we contain a bunch of atlases and segmented volumes. We have the MNI 111 or 1 millimeter segmented volume, which is what a majority of all of our light modeling tutorials use.
Okay.
So you can go in the toolbox for that. You could also search online for other ones. But if you're doing adult, the MNI 1 millimeter segmented volume should be pretty solid enough. We also have segmented volumes for younger children. I believe we have like 6 months, and then 3 months, 1 month, and I think we might also have 6 years.
Okay,
as
and
well.
you said that the 1 millimeter, the common one,
Yeah,
is
the MNI
in.
is in the toolbox. So
Toolbox.
I can show you where that is in my file directory.
Thank you.
So I will head down to my MATLAB folder. So in NeuroDOT, we have data, documentation, functions, and support file. All of our sample data is in data. All of our like PowerPoints and tutorials are in documentation. Functions contain the actual code that you guys use. And then support files is where we have all of our extra tools. So A matrices, atlases is what you're going to look for. So inside atlases, we have masks, we have atlases, and then we have nifty files for a bunch of volumes.
Okay.
And so you can just load in whichever one you need to for your segmented volume. So right here, we have a segmented volume for a 1 millimeter with like extended boundaries, which is useful for if you're scaling the mesh up to larger head sizes like myself. We also have the normal 111, which is down here, and then we have it with 3 millimeter voxelations instead, which is just a larger voxel size.
Perfect. And then so that name is what you would put in the first area.
Exactly.
And then what would you put in the pad?
The pad file would be whatever pad file you create for your own system.
Okay.
So for that,
And that's what we did up there.
That's what we did at the beginning of the script. What you would end up doing is loading in your DOT data,
Okay.
extracting the 2D and 3D optode positions, which
And-
would be a little bit different than what this is because this is using a NeuroDOT data sample.
And the DOT data is probably what we did for the pre-processing?
Yeah.
Okay.
So if you have like a SNRF file or a NIRS file from your system that you're using, you would take that, find the optode positions in, for example, the SD structure for NIRS or in the probe structure for SNRF, and then just extract those positions, put them into a grid structure like in lines 48 through 50,
Perfect.
and then build your pad file from there.
Perfect. And so whatever name we give it there is what we put down below.
Absolutely.
Okay. Sorry for all the questions, but
Oh.
thank you.
You're asking great questions, no problem at all. We have a question in the back as well.
Thanks. So once you've worked out like the X, Y, and Z coordinates of where you want your pad generally to be on the actual array, can you
Yeah.
just have that like automatically there, like preset that, and then whenever you want to like, if you have the different measurements for the people like Nazi and Daniel and whatever, can you just like run that automatically through to generate like the A matrix for each participant without having to go into the like align me?
That's a great question. You could certainly, you would probably need to hardwire your own code for that, to do that. I've built a pipeline for a collaborator that has done similar things. So if you wanted to work with us on that, or if you wanted to do it yourself, that is 100% possible and doable.
Okay. Yeah, no, that sounds great. Thank you.
Awesome. Yeah. You can see it took around 5 minutes or so for wavelength 1, so we got probably another 3 to 5 minutes left for wavelength 2, and then we can move on. And so another notable thing about the light modeling process that I have yet to mention is we started with a 1 millimeter volume to create our mesh. But when we actually create the A matrix itself, we use a voxelation resolution of 2 millimeters instead. And that helps with file size, because what we're doing here is for every individual source and detector pair possible, we're creating a Dreens function, multiplying those together, and calculating our sensitivity for that measurement. So, and that's through a set of voxels. So if you can imagine having one of our fiber-based systems that has 125 sources and 128 detectors, that ends up into the tens of thousands of measurements. And if you're doing that over 1 millimeter voxels, your file size will get pretty large. So doing a 2 millimeter resolution A matrix is a way to have a little bit of a middle ground. You can create it on 3 millimeters as well, or you can create it on 1 millimeter if you want that high resolution. But now that perfect timing, this is our simulated light fall off. I built this pipeline so that way when you've finished generating the A matrix, this figure pops up, so you have just another visual cue that, okay, I can move on to another step in this pipeline. And remember what Adam was saying earlier, where this simulated light fall off does not include any noise. So it is an idealized version that's not fully accurate to what you're seeing in your data. So now that we've made our A matrix, we can just do a little bit of a reshaping, package some info together, and then save it. So I'll run that right here. Oh, I'm trying to save into the wrong place. But, oh no, I'm saving in the right spot. We're good. So 2 variables that get saved out when you generate the A matrix are these gfunc variables. So you have them for, they get saved out within the A matrix generation function. And then we save our A matrix right here. And now we can visualize aspects of our sensitivity profile. So we want to take our volume that we're visualizing on, put it into the same space of the A matrix. So that's what this first line does. And then because we're going to be visualizing some single source detector pairs, I'm going to pull up a 2D plot of the pad. So I've included some base, easy source detector pairs. But I'm going to turn this over to you guys and pick on some people now. Someone in the audience, give me a source detector pair that we should visualize. I'm going to choose, let's go Caitlin over there. 1 to 7, so that'll be source 1 and then detector 7 right here. So I'll scroll down to right here for the try your own. We'll set 1, and then we'll set 7. I need to remove the quotations, otherwise it won't work. And so what we can do here is see our very large banana from source 1 to detector 7. Alrighty. Thank you, Caitlin, for that. We'll move on to the FFR. So flat field reconstruction, we do a quick reconstruction pipeline. You can see our Tikhonov invert A matrix function right here. We smooth it, and then we make our flat field reconstruction. This will take probably 2-ish minutes. Oh, no, that was a lot faster. I lied. And so now we can see our sensitivity of the system. We can see how it looks on my head, what this pad sensitivity would have if it was actually on my head in the lab. So we can just play around with that and then close out. And so our last little bit that I like to do is just putting everything together, running this final section so we can see a fun little visualization. And so what we get here is my head with a brain inside, and then the positions of all our sources and detectors on the back of it. And we get a nice little view of putting all of our pieces together at the very, very end. And with that, I'm going to say thank you all for having me here. Oh, we do have one more question?
Yeah.
From Zoom.
From Zoom, someone asked, why did you choose to have more sources than detectors instead of the other way around? And is it because of cost or something else?
That is a really good question. Unfortunately, I was not here when that system was built. So can I throw this to you, Joe?
I have an answer.
Thank you.
So first, there's almost identical numbers of sources and detectors, right? They're sort of subtle, like maybe 24 versus 28 or something like that. What we do is we actually usually don't completely fill the detector channels because we want one of the detector channels to be the sync signal. So that's why we would drop down on that. But otherwise, I mean, we're trying to make it as symmetric as possible. That's what, 25 versus, no, 28 versus 24?
Yeah.
Yeah, so I mean, we keep things as symmetric as possible.
And for those of you here who may have been a little bit confused what Joe meant by sync signal, that is how we synchronize our events for the stimulus with our actual data. There are multiple ways to do that. You could do it with like a TTL pulse from a cable. You could do it with just like an audio boop that like is played and recorded in the data. But there's multiple ways to like synchronize your data with your stimulus. Yeah, I'm going to hand it over to Vita now, and she's going to talk about validation using fMRI data.